finding certain structures, they just sort of go to a dead end in some cases, and I find that fascinating. But what we're looking at here, of course, this thing you can turn around and rotate in various ways. You can see the three-dimensional relationship. But we're looking here, this is the basal nuclei, this is the putamen. And then peripheral to that is the caudate, this one's tail. And so you can see in 3 dimensions, but you don't see in the slices. You don't see in the slices, you don't see this C-shaped conformation. And then here's the globus pallidus. But remember, this constitutes the lentiform nucleus and most of the central structures of the basal nucleus. Again, you don't often. And then if you hide these, you can see the relationship of these structures to the, for example, the hypothalamus. There are the ventricles there. Hide that. You can see the corpus callosum there. They're showing you the choroid there. I think that's kind of silly. But just a wonderful little tool. And then, of course, we talked about the limbic system. There's your amygdala. Hope you'll take advantage of this.
What is this? Which part of this?
Oh, as we've been talking, we began our discussion of the central nervous system, looking at the brainstem and the thalamus and the basal nuclei, cerebellum, and associated structures. Remember in Paul McLuhan's discussion, those structures represented the reptilian brain. The reptilian brain, your brainstem, and then associated with that were the paleo-mammalian structures, which, for the most part, in his opinion, constituted the limbic system. I don't know if that's a higher system or not. One of the problems, neuroanatomically or perhaps neurobehaviorally, is that the limbic system and the higher cortex, the human brain, the higher cognitive region we're talking about now, they don't communicate all that well. And most of the communications that exist are one way. Remember, we talk about thalamocortical shaft, the thalamus and the cortex are always talking, exchanging ideas. Well, if you think about what happens when you get angry, you get all riled up, festered, deceiving, and so on. And if you probably recognize, rationally, you recognize, hey, this isn't very productive. I'm in a situation where I can no longer be angry. I need to calm down. But that's a much more difficult thing to achieve than might be desirable. And that is in part because of the limited connectivity between the rational brain and these limbic regions. The limbic regions are quick to go to, the term that we use to describe the onset of an epileptiform seizure. We're going to go to the term kindling. And there's a similar analogy with anger, don't you think? Don't you think that sometimes it's like you start the fire with kindling, but it's once you've got it going, it's a little hard to dampen. My whole point here is that I just wanted you maybe to reflect upon the neuroanatomical relationship. So we're talking about the neocortex, and we talked about the relative localization of function. For example, we said the anterior brain is the motor brain, however abstractly defined, and then the posterior brain, in many respects, is the sensory brain. And in fact, you have visual centers, auditory centers, you have tactile centers, and we sort of integrate everything in this PDO region, the parieto-temporal occipital cortex. So that's the general thrust of these. We talked a little bit about the consequences of eliminating the prefrontal cortices through a lobotomy. We talked about what would happen to such individuals if suddenly bereft of their ability to anticipate the consequences of their actions, if they no longer have the ability to perhaps weigh right and wrong on a variety of levels. But the other thing, and I guess this, if we have other images here, maybe we can use this one. I guess this one's a little better because it's a little larger. I just wanted to make sure we understand the relationship between, because we don't have time to get into this in great detail. The primary cortices, the primary motor and sensory, and the association or supplemental cortices. So let's use use the anterior cortex. Here's the central sulcus. So this is our primary motor cortex, or primary motor strip. And we've already, its function is fairly clear. What is the function? What would you say is the function of the primary motor strip? You're explaining this to a fifth grader. What are you going to tell them? What is the primary motor strip do? All right, good. Well, the primary motor strip includes the neurons that are immediately responsible, probably the better word is penultimately responsible for contracting a muscle. The primary motor cortex, within the primary motor cortex are the upper motor neurons, that extend from the motor cortex down to the spinal cord. And then from there will synapse with another motor neuron that goes from the cord to the muscle. So as we'll see in just a moment, the motor pathway, the corticospinal pathway, for the most part, consists of two neurons. So that's why this is important. And then what we're saying is that, in a sense then, ideation, motor planning, and motor execution sort of moves in this direction, right? It moves from anterior brain back to the primary. And so all of these association and supplemental cortices are simply refining, simply dealing with the details of the execution. This is called the final common pathway. This and the lower motor neuron. Now, with respect to the sensory brain back here, of course, it's a little more cosmic. It's a little more difficult. But in general, I would suggest that it's just the opposite. That initial sensations, I have to be careful here because, of course, the regions back here are highly localized. But in general, we can say that at each lobe, each primary sensory cortex, that is where the sensations are initially received and processed. So the sensations are initially, so it's kind of backwards, kind of opposite from the motor, are initially received in the primary cortices, the primary sensory cortex. So for simplicity, we'll just call this the primary sensory strip or the primary sensory cortex to keep things simple. And then as we move posterior from this strip, progressive integration is going on. You're integrating tactile senses with auditory senses, with visual senses, and so on. And as we'll talk about in a moment, as all of that is going on, and you're doing all of this processing with the sensory information, you're drawing upon association, you're drawing upon memories, you're drawing upon emotional tones from acquired or stored in the limbic system. So all of that is going on back here. So imagine running a computer program just to vaguely simulate any aspect of this. It's just mind-boggling. So that's the general theme here. Again, when we talk about association and supplemental cortices, we don't have time to get into the details, but that's the general organization, both in the motor brain and the sensory brain. And then bear in mind as well, while all of that's happening, there is, to a greater extent than in the anterior brain, a localization of function. We have the visual cortex back here, the auditory cortex back here, the somatosensory cortex in this region, integration in this region. Now, we also talked about somatotopic organization, especially of these two regions. And what do we mean by that? What was somatotopic organization? Exactly. We'll come back to that. So Bob said, like the homunculus, and I have a picture of the homunculus coming up. For example, on the test, I might ask you, on the next physiology test, I might ask you in a very, very general sense, what region you have a patient who suffered a stroke, they no longer have sensations in their feet. So I might either give you a picture or ask you in very simple verbal terms where that stroke is most likely to have occurred. And we'll come back, we'll look at that illustration, will be coming up here, in fact. So, according to our somatosensory homunculus, if on the one side we have a sensory homunculus, and on the other we have a motor homunculus, this would be the sensory here, this would be the motor, so they're very similar. But where would you suspect initially that the brain had undergone hypoxia? as a result of the stroke if they're no longer sensing, if they're no longer receiving sensations from the feet. What do we call this region down in here? It was the longitudinal fissure. Remember the longitudinal fissure? So you would say it's deep within the longitudinal fissure. I just want you to be able to verbalize these in very general terms right now. And because it's sensory, you would tell me that it's related to the post-central gyrus, or at least be familiar with all this terminology. Post-central gyrus, primary sensory strip, something like that. And then you can see, for example, if the person is moving their tongue there, let's say as a consequence of some type of cerebral trauma or stroke or whatever, they lose the ability to move their tongue. What region of the brain would you describe that, or would you ascribe that to? Based upon our discussion of the lobes. Yeah, you're probably somewhere in the temporal lobe, or the anterior frontal lobe, or lateral frontal lobe. But yeah, where this is set, where this is worked out, this would be a temporal phenomenon. The pathways are a little more complicated.
Oh,
yeah.
Go first.
I was going to ask, so with the motor primary strip, it's on both hemispheres, right?
Yes, it's going to be on both hemispheres, left and right. We're going to talk about laterality of the brain in just a moment. But yeah, exactly. Yeah.
I was going to ask, what you said confused me a little. Does the central sulcus extend into the temporal lobe? I'm sorry, the central gyrus extend into the temporal lobe.
Well, the central sulcus would extend into the.
Because that's the lateral.
Yeah. That's why I say it's a little more complicated with pathways. The temporal lobe, a motor dysfunction, as it turns out, as you follow that motor strip down, it carries on to some extent in the temporal lobe, such that an infarct in the temporal lobe or in the about the only way you can characterize this is in the lateral motor cortex, the lateral motor cortex, or the temporal lobe, and in both cases you would have a loss of motor. Yes.
So, talking about your question with the loss of ability to movement of the tongue, lateral motor cortex would be like the sufficient answer to that, or the sufficient answer is temporal lobe. I guess I'm confused on what.
Yeah.
It's part of the motor cortex, so I guess that's where.
Yeah. The problem is that this is in both cases, in both the sensory and the motor, this is not a terribly well-defined strip, not a terribly well-defined region. We're calling this an area. All right. It's not as if there's a single sulcus that goes down along the strip there. It's an area. And what we're saying is that area includes the frontal cortex, includes the frontal brain anterior to the central sulcus, but it also includes portions of the temporal lobe as well. And it's a little more ill-defined, and it changes shape there. But the somatotopic organization still continues. So, yes, you would, obviously, this is not the frontal brain here. Right? So, don't ascribe at this level too much precision to this. They're showing this as a single gyrus here. There's no single gyrus that corresponds to that area of strip. Now we're looking. I think this is actually a cat cortex, but it would apply to our brains as well. I've applied for a cat cortex transplant because I think it might be useful in some respects. I learned today, for example, that if you want to know how to make your patients happy, there have been several studies, published studies, showing that all you need to do is show them cat videos. Cat videos make us all happier. Don't really know why that is. Now we want to talk a little bit about the stratification of cells in the cortex. We often, or in the anatomical literature, you'll see references to the neocortex and the allocortex. The neocortex is characterized by the presence of 6 histological tissue layers, I guess. And they're numbered from 1 on the outside to 6 on the inside, of E. And you can see some of that laminar organization here. This would be layer 1 here. This would be layer 2, 3, and then it gets a little fuzzy, but 4 and 5 would be down in here. We don't really see 6 all that clearly. But let's go back and look at this illustration. You'll notice layer 1 consists primarily of interconnections, primarily of dendritic horizontal connections between adjacent regions of the cortex. There are a few cells up there, usually small. Some of them are called horizontal cells because they extend these horizontal processes, but primarily a region of intercommunication. In layer 2, you'll notice here, there are relatively few cells, but what you have are a lot of ascending and descending processes. So mostly ascending and descending fibers. Relatively few cells. One of the patterns, of course this is going to vary from region of the cortex to one region to another. One of the general patterns is that as we go from layer 1 to layer 5, the size of the neurons tends to increase. The size of the neurons tends to increase. And in general, in most parts of the cortex, the number or density of neurons also increases between layers 1 and 5. So they increase in number, increase in size, but they also increase in, they change. They change in function as well. And so as we've indicated, relatively few cells, if any, in layer 1, relatively few cells in layer 2, but in layer 1 you have horizontal connections, in layer 2 you have predominantly vertical connections. Then in layer 3, in most regions of the cortex, you have small, what are called projection neurons. You'll see at several points here the letter P. The P can stand for projection or pyramidal. The terms refer to 2 different aspects of pretty much the same thing. So what we're saying is that pyramidal neurons in layer 3 tend to be usually smaller than pyramidal neurons in layer 5. And we call these projection neurons, you don't have to pay attention to the recurrence, but you see that some of these give off recurrence to other cells. We call these projection neurons because they, like the Purkinje cells that we saw in the cerebellum, are aggregating information from a variety of neighboring regions and then sending that by their one axon to some other portion of the nervous system. Could be into the white matter and going to another region of the cortex. So the general rule is that it's going to vary, because the function of these cells is going to vary depending on whether we're talking about the motor cortex or the sensory cortex. But the general rule is there's the greatest abundance of cells in layers 3, 4, and 5. 1 and 2 is relatively sparse. And then 6 consists primarily of these projection axons. And then down here, we're in the white matter. And you see something of that in this illustration here. Again, relatively sparse distribution of neurons, but it picks up as we move deeper. And then you'll also notice the size of the neurons tends to increase. These letters, this would be called a fusiform cell. It has a sort of tapered shape. There are cells of, what is it called, Marinahi, usually 5 and 6, not terribly important. S often stands for stellate cells. We've learned a lot about stellate cells. We think that stellate cells are very important in higher cognitive functions. Probably the one you should know, that I should mention here, is the B here, which is bets. Bets. Not what you do on weekends, but there's a Z at the end. That's used to be a great guitarist for them. And these, in the primary motor cortex, are the projection neurons. They don't show it very well here, but these are the projection neurons that are responsible for the corticospinal tract.
I'm sorry, what was the responsibility?
The Betz cells in the deeper layers of the primary motor cortex are the upper motor neurons. These are the neurons that send the signals to the spinal neurons that are responsible for contraction of muscle.
Do they do it through the projection cell intermediary?
No, as I say, this is, I don't know what part of this illustration, but this illustration could refer to any part of the cortex, and on all part, this could be the sensory cortex for all I know. But Betz cells have a shape more like this. They are standard projection of pyramidal neurons in the primary motor strip. But you can see even the Betz cell in this illustration is a fairly large neuron. But these are the classic projection of pyramidal neurons. So we've been talking about these projection neurons and the way that they send their efferents, their axons, to other regions of the brain. Let's talk a little bit about the organization of these wiring systems, the organization of the white matter and the cerebrum. And there are three general categories, three general types of classes of white matter pathway in the brain, and they're fairly logical. For example, you can see what we've been talking about. These long black lines here are the projection pathways that are responsible for, as you can see, interconnecting the brain and the body, the cerebrum and the body. That would be true for both motor and sensory function. You'll note that they decussate, they originate, you don't see all that well here, but they originate up in the cortex. Humans, the cortex is only a few millimeters thick. Usually it ranges from 1 to 4 to 5 millimeters thick, most commonly around 2 or 3 millimeters, so it's very, very thin. And we call this decussation. In this case, you can see it occurring for the most part in the brainstem. So the projection neurons connect the brain and the body. These blue fibers that you see here are usually called commissural or callosal fibers, commissural or callosal, and their function is to interconnect the two hemispheres. Corpus callosum is the largest of these connections, but there are a couple of others associated with the corpus callosum. There's a lot of debate now about the differences in the male and the female brain, and there's pretty much common knowledge in the reading the textbooks that the female corpus callosum is larger than in the male. What we found out is that there's more white matter in the female brain than the male brain. And so does that mean anything? And I guess depending, now all these answers depend upon your political views, so I hesitate to even offer an observation on the topic. But the previous consensus was that females were better multitaskers than males. The problem with that is we have learned a lot of things about multitasking in recent years. How many of you believe yourselves to be good multitaskers? You do lots of things simultaneously. But the rest of you?
No.
No? That kind of destroys the point I was going to make, because a lot of the research shows that individuals who do the actual studies, individuals who think that they're good multitaskers usually are not all that good. And a lot of those who don't think they are do better. The real problem, the real issue, though, is that the multitasking, and who's not doing it nowadays? I see somebody on the cell phones in class. I see all kinds of things. We're driving, we're talking, we're all texting when we're driving. Right? We're all doing multiple things, but there's a lot of evidence to suggest that it's not good for your brain. It's not good for your ability to concentrate. In fact, there are some neuroscientists who believe that daily multitasking causes irreversible brain damage and compromises your ability to focus and concentrate. In any case, we'll talk more about these commissural fibers, and there are individuals, they're very rare, individuals who are deemed acallosal. What would that mean? Acallosal.
They don't have connection?
Yeah, they lack the normal radiation of connections between the two hemispheres. And as we're going to see, the two hemispheres have a slightly different function, and they're communicating with each other. So, in such individuals and in individuals who have had the surgery for epilepsy, where maybe the two hemispheres have been separated, why would they do that? Why would they Why would they sever callosal connections in epilepsy patients? I'm sorry? Yeah, I think you're right. The answer was too much fire. And what tends to happen, we talked about the kindling. It'll start, Hewlings Jackson, the great British neuroscientist of the turn of the 20th century, talked about the grand march. And it'll start somewhere in the body and then work its way up, and then a portion of the entire body is involved. And then eventually you have both halves of the body involved. So the idea is that you're going to stop that. You minimize the progression. It's kind of a blunt therapy for a very subtle disorder. The other class, the last class of white matter pathway we should worry about or should mention are these association fibers. And they're kind of hard to see here, but they're the little red regions that you see there. The association fibers generally act to connect or interconnect the motor brain with the sensory brain. So they sort of go fore and aft within each hemisphere. Some neuroscientists speculate that that's the seat of your consciousness. So we have association, commissural, projection fibers, just to be aware. We talked about the connectome. Here's the corpus callosum. You can see how large it is. Just a massive band of interhemispheric fibers. That's some products of the Human Connectome Project. And as I said, when I look at this, I'm just struck by the remarkable linearity and organization of these white matter pathways. And you can see, I mean, what pathways are these? The projection, what is that? Yeah, that's the commissural or callosal pathways. The software that is able to do this is readily available. You can download free software now that if you have the proper CT scans, and that's not a big deal either, the proper CT or MRI, you can take this diffusion tensor software and actually render these. Of course, the coloration is later. But you can see this, again, I'm struck when I look at these by the regularity, the order, the organization, and the white matter pathways. We're looking at here, and we've seen several images already of the way that the cortex is perfused by the cerebral arteries and veins and so on. Remember, for the most part, we looked at the vertebral arteries, we looked at the carotid, and we saw the anterior vertebral in the middle, the anterior cerebral and the middle cerebral and so on. And we saw that in most cases, those vessels seem to only supply the surface of the brain. Well, in part, that's because that's all the action is. That's where the cortex is performing most or burning most of the energy. But what we're looking at here is these are the vessels at the surface of the brain here, and this is how they radiate more deeply into the cortex. And this is the perfusion of capillaries that is responsible for energizing, supplying, feeding.
Are those arteries that you circled on the top, are those in the subarachnoid space?
Right, we didn't, but you remember when we looked at those callosal and anterior cerebral arteries, how they seem to come up the margins of the longitudinal hemisphere. And then you have a radiation of vessels at the surface of the brain. So all we're looking at here are the efferents, the little tributaries of those vessels as they enter the cortex.
Dr. King?
Yeah.
So do they descend into the sulci, or it's actually they go inside of the white matter?
No, well, the larger vessels are going to be more abundant in the sulci, but remember, sulci is just a structural. It has no significance except that if you just take our brain, if you take the carpet here and we want to move it to a smaller area, we're going to have to get it to fold. 2/3 of the surface of your brain are within these tissues, these sulci. So all of that region has to be perfused, just like what you see on the outside. So it really doesn't make a lot of difference. In some cases, the larger vessels do enter through those folds, but you're going to see this for every portion of the cortex. All right, we've already talked about it. Just trying to finish a few simple points. So we've talked about the way that motor commands are handled in general in the brain. We said there are 3 primary centers that regulate, control motor function. We said that it is the primary motor cortex that is responsible for, well, it's the final common pathway. It's the upper motor neuron that's sending that projection neuron up there in the cortex is sending the signal down to the lower motor neuron, which in turn is going to be responsible for muscle contraction. All right. We also said that as that's occurring, some of that information is being bled off to the cerebellum. The cerebellum is then taking sensory information and monitoring the motor efferents and making sure that those are all consistent. It's taking your volition, remember your volition is your will, your intention, and comparing that to what is being perceived by the tactile system, the visual system, and so on, and making corrections on the fly. And then the basal nuclei, we said, is more of a centralized efficiency system. It's an inhibiting system. I know that I was not a very gifted athlete. I tried awfully hard, but I was never better than mediocre. I remember going to a karate session one time, and I just didn't beat up awful, just really bad. But one of the problems I had was when I would try to block a blow from the 4-year-old who was beating me up. The arm wouldn't stop. It would just keep going. I would try to block it, but in my effort, I guess, again, because of impaired motor function, the arm would go way too far. So my point is, the basal nuclei in most of us tends to inhibit or prevent that excess exaggerated motion in addition to the pathological movements that you'll see in patients with various types of tremor, palsy, athetosis, melismus, chorea. I mean, you're going to learn a whole list of motor control problems. In many cases, not all, but in many cases, they are attributable, at least in part, to a deficiency or breakdown in this overall inhibition system. So, and that's what this little diagram here is trying to suggest. The basal nuclei, for the most part, tend to inhibit undesired or unwanted gross motor movements. The cerebellum, in an excitatory fashion, pushing, nudging in a very gentle way, is correcting especially fine motor movements from the corticospinal tract. And so what we're looking at here, these red lines represent, represent the corticospinal tract. And remember, it pretty much consists of 2 neurons, at least in our very basic discussion, consists of 2 neurons, an upper motor neuron and a lower motor neuron. So we can start over here in this illustration. What are these red things here? What are those red things? They're not arrows. That's why I put not arrows up there. I don't want you to think that the signal is going this way. It's not. These? Yeah, those are cell bodies of those projection or motor neurons. That's going to be the cell body of the UMN. What is the UMN? Upper motor neuron. Upper motor neuron. And so what do you notice about it? On the right side here, it's descending on the right side until it gets to the pyramids of the medulla, the medullary pyramids. We have crossing over or decussation. About 75–80% of the corticospinal fibers cross over in this way. Others do more creative things. All right, let's follow one down here. It's ongoing. I don't see any breakage. It continues. And then what is this? That's the synapse between the upper motor neuron and the lower motor neuron. And so this neuron, located in the ventral horn of the cord, wherever it is then going to send out the signal, the efferent signal, the motor signal to induce motor contraction.
It's
And
postsynaptic motor signal.
upper motor neuron, lower motor neuron. And you will study upper motor neuron diseases and lower motor neuron diseases. In Australia, they call Lou Gehrig's disease lower motor neuron disease. Although that's not.
So the projection neuron's axon will synapse with the cell body in the ventral horn on the contralateral side.
Yes, on the contralateral side. This is true for the corticospinal tract. Again, about 75, 80, 85% of the time that's the way we'll work. So that's all we're looking at here at both pathways. I just want you to pay attention again, make sure you understand this distinction between the upper and the lower motor neurons. This is kind of a little silly illustration, but I just sort of want to put some of these things together we've been talking about. The image here is intended to suggest the sequence of brain regions that must be called upon to what do we ask you to speak a heard word? Speak a heard word. So we're going to find out this is the temporal lobe. Most of auditory processing occurs here. So the information is going to end up in what's called the auditory cortex. As we'll see, for most right-handed individuals, the language function resides in your left hemisphere, more specifically your left temporal, and to a lesser extent the motor cortex, but it's on the left side. So this auditory cortex is basically it's basically processing the sound. It's just basically trying to figure out what sounds are important. You have a remarkable ability. If you're at a party, in fact, it's sometimes called the cocktail party effect. You have the ability to, if one of you starts talking in here, and then maybe that excites the rest of you, you start chattering too, but nevertheless, you're able to focus on that one person and hear what they're saying. And if you're listening to a symphony, in many cases, you can stare at the principal violinist and actually pick up what they're doing. You're using motor information, all kinds of information to do that. All right, that's in part what this region of the brain is doing. And then those basic sounds, which are just sounds at this point, perhaps even noise, are then conveyed to Wernicke's area. Wernicke's area, which you can see is right there beside it. And Wernicke's area is the region of the brain, temporal cortex, that is your speech processing center. It takes those sounds and then converts them into meaningful language. It's this part of the brain that's thicker if you're multilingual. Now remember, our job is to speak something we've heard. So all we've done now is converted those sounds into languages.
Does it affect what area of that does it affect?
So the question was about aphasia, and there are many different types of aphasia. Aphasia can result, yes, it could result from a problem with the ability to interpret sound generally. It could be a problem with the ability to convert those sounds into language, or because it's such a general term, you'll learn about different types of aphasia. We'll use a very clinical precise designation. It could be in some of these other circuits that we're talking. In other words, you're going to have patients that understand everything you say to them. They can't respond. Or when they try to respond, Tuesday, burger, shoes, foot, it's word salad. They think they're saying something meaningful, and they get frustrated that you don't understand what Tuesday, burger, foot means. So this is the lingual conversion area, and as you can see now, we're going from the temporal cortex up to what part of the brain is this? Frontal or motor region. And so now this is going to take this lingual information and now convert that into a series of motor procedures, a series of motor routines that enable you to move your tongue and your lips and your mandible and so on in a perfectly correct way. So this is the speech articulation center. But we're still not finished, because even though those motor routines are all set and ready to go, you know how to pronounce ping pong, and you're ready to say ping pong. In order to say ping pong, where does that signal have to go? To the motor cortex.
You have to go through the cerebellum, it's the second line.
Yeah, all of these would go through the. Yeah. Again, we can talk about the olfactory cortex and the balance, the vestibular processes. But there are very few exceptions. I think if we just sat down and we had time to talk about this, you could see that virtually everything, every enterprise, every activity that you're involved in involves an immense coordination of neurosensory and motor functions. Yeah. It could vary, but usually if it's word salad, the implication is that their intent is formed. They know what they want to say, they just can't say it. So usually it's Broca's area. Carl Sagan used to talk about Broca's brain. He had Broca's brain in a jar by his desk. So that's it's actually much more complicated than that. But here we're speaking a written word, similar sort of thing. You're going to take the visual information into the eyes. It's going to go back to the visual cortex, along with some other regions. You should be able to tell me where else this information is going at this point, but this is what's important for this task at hand. Going back to the occipital, the primary visual and secondary visual centers for interpretation. We'll talk more about that later. And then, again, because our purpose is to speak a written word, it has to go to the language processing center, because really what is written is just a series of lines, or if it's a different type of language, symbols. I mean, how on earth do we make any sense out of just a bunch of horizontal lines and vertical lines and a certain relationship? It's almost magic. But in any case, and then from there, it goes to Broca's for the speech routines, and then back to the motor cortex. Yeah.
Is there the assumption in some of these speed reading theories that it sort of maybe doesn't follow the same pathway?
I don't know. There's so many of these speed reading things out there. I'm not sure which one you're talking about. There's a lot of these apps that you can buy now that will just show you one word at a time very rapidly. Is that what you're talking about? Or are you talking about the phrase, you read a line or a phrase at a time?
Yeah, kind of take chunks.
Yeah, I don't see any, in that school, I don't see any great deviation from this. And then what I was going to say is a lot of those apps that came through, and I actually know one of the fellows who's making money off this, selling one of these for an Apple. I don't know that the science has supported the fact that it actually speeds up your reading, or at least not your comprehension, because we all probably read 1,000 words per minute. We don't have to pay attention to, we don't have to comprehend what we're reading. But it's the comprehension, and the comprehension tests for some of these seem to suggest that it's more of a natural facility that you can develop. But in any way, getting back to your question, I don't know, I'm not aware of any deviation from this. What would you suspect? Do you have a theory, or are you?
Well, if you're not having to sound out the whole word, maybe it's.
Oh, I see. And you're saying that. Well, in the reading, all you're doing is just getting to this point anyway, right?
Yeah, I mean, sounding it out.
Oh, I see. Okay. You may be right. As I say, all of this is a lot more complicated than these silly, simple illustrations. All right, let's take a break and we'll